Okay. Hi folks, Chris Summers here from the University of Regina, Department of Biology, perhaps better known as the founder of the Saskatchewan Sportfish Research Group. Out here on Buffalo Pound Lake today, doing some field work, tagging fish, trying to understand more about fish biology in our province. It's a big focus of our group, but perhaps what's most important to me is the opportunity to work with a lot of enthusiastic young people who are the future of our fisheries and wildlife sciences here in Saskatchewan, and mostly I'm going to let them tell you about the research activities we have ongoing. It's been a weird year. Pandemic has changed a lot of our lives, and of course we don't have the opportunity to come talk to you in person, which we love to do, but we still want you to know that we're out here and we are working hard to improve knowledge about our fisheries. And we are working in a number of specific areas. We do work on catch and release biology, invasive species, fish habitat use, space use, and we really are trying to gather a lot of information that's relevant to long-term management and conservation. I'm going to update you a little bit today about our T-bar tagging program, and I'm going to let my students tell you about the rest of the work that we're doing. So we have been tagging fish in Saskatchewan water bodies since 2017, and we're about to reach a major milestone, which is that we have almost 10,000 fish tagged over that time period. And it's a really unique tagging data set because almost all of those fish come from angling, right? So there's not a lot of fish in there that come from standardized fisheries sampling like you would see in other jurisdictions. All of those thousands and thousands of fish have been caught and released by anglers. And that really gives us a unique opportunity to see what happens to fish, how they move, what habitat they use, and what features contribute to successful catch and release. Now, one of my favorite aspects of that program is the citizen science component. We tag the fish, but it's anglers who report them back to us and contribute the data to the long-term data set. And that opens up a line of communication between scientists and anglers that's really unprecedented in Saskatchewan. And we really appreciate the involvement of anglers in that process. Okay, so how do you report a tagged fish? The fish has a tag that's going to be on its back near a dorsal fin. Usually it's blue, yellow, red, or green in color. And on that tag is going to be information about how to report. There'll be an email address. You can send an email with the information about where you caught the fish, the date, and any particulars about whether you kept the fish or released it. And importantly, there's both an email address and a unique ID number on the tag. And the ID number links that fish to a specific record in our database. So when you report, if you could please report the ID number as well as the rest of the information about the fish, that would be fantastic. Now, we've received over 1,000 reports of recaptured fish so far, and we are learning some really cool and interesting things. First off is that fish are way more mobile. We're talking about walleye, for example. Fish that we've tagged in the central part of Lake Diefenbaker show up close to the Alberta border. Right? And fish that we tag in Last Mountain Lake show up in Pasqua Lake near Fort Capel. And so we're seeing fish moving and using space and habitat in ways that we really haven't had insight into before. One of the most important things that we're finding, and one of the most interesting in my opinion, is that catch and release is supposed to be a strategy for conservation and management. We want to have a chance to enjoy fish, but then put them back in the water for other people to enjoy and to have them contribute to the fishery long term. Our tagged fish are showing that not only is that happening once, but we're having fish that are caught twice, 3 times, 4 times, even 5 times on Lake Diefenbaker and on Tobin Lake. So yeah, our tagging program is really showing us how catch and release can be effective in real time, right before our eyes. It's not all great and good news though. There are some challenges that we see with fishing, and we want to keep working with important stakeholders like the Saskatchewan Wildlife Federation to help improve in these areas. And probably the most important one that I want to mention is something called barotrauma. And barotrauma is damage that fish sustain when they are caught in deep water and brought rapidly to the surface. And this is particularly important for fish like walleye and yellow perch, and also burbot. Really the only way to avoid it is not to fish in deep water. And there are some relief methods out there that people use, but we don't really know if they're effective or not. And so one of the things we'd like to do moving forward in the future is to learn a lot more about barotrauma and how to deal with that potential issue. So stay tuned, we have a lot more research coming up. We really miss the opportunity to interact with the Wildlife Federation, Saskatchewan Wildlife Federation in person, and we hope that 2021 is going to be a better pandemic-free year, and that we'll see you out on the lake at some point and at the convention in 2022.
Hi, my name is Carmen Kotowicz and I'm from the University of Regina. I am a brand new master's student of biology starting this semester, January 2021. My research is on round and lake whitefish in Lake Huron, and I am studying the population structure of these fish using stable isotopes. Stable isotopes can help to determine the feeding ecology of lake whitefish and round whitefish, which can help to determine their different populations. Figuring out the populations of these fish is important when considering different management practices. In Saskatchewan, we have large bodies of water with lake whitefish in them. Therefore, understanding the best ways to determine the population structure of these fish would help to inform management strategies.
My name's Ryder Rutko. I'm a master's student at the University of Regina. I'm originally from Mossbank, Saskatchewan. And today I'm going to talk to you a little bit about what my project is and what I've been up to lately. So, I do research on yellow perch in Lake Huron, using life history traits of yellow perch to look at population subdivision and the effects of latitude, longitude, and depth on yellow perch life history. So my research focuses on yellow perch, and yellow perch are actually a much more important species than a lot of people give them credit for. So they play a super large role in both the recreational and commercial fisheries all over Canada, but particularly the recreational fishery in Saskatchewan. So yellow perch is a species often targeted by anglers in both the summer and winter, but particularly in the winter. So understanding stock structure in different lakes, especially in regards to yellow perch, would be super critical for proper management of perch populations in various lakes around Saskatchewan. Yellow perch are also a very critical prey species for a lot of fish. So big walleyes, big northern pike, they eat little yellow perch. So it's another good reason to try to preserve yellow perch populations and understand where they're located so we can properly manage for yellow perch populations. So both a very important recreational fish species, they're well sought after by many people, and a very important prey species as well. So I'm in the final year of my master's project at the University of Regina, and I'm really excited to pull everything together here, get a paper published, and actually share with you guys all my results from my yellow perch population study. So I'm hoping to bring that to the Saskatchewan Wildlife Convention in 2022 when we can finally all reconvene, and I'm really excited to share with you all what I've found. So thanks.
Hi everyone, I'm Shana Hamilton, and thank you guys so much for allowing me to speak again at your convention. You've probably seen my face a couple times if you've been here before, and this is currently my third year of my master's at the University of Regina with the Saskatchewan Sportfish Group. I wanted to give you guys a huge thank you at the Saskatchewan Wildlife Federation, because without your guys' help, my project would not have been able to happen how it did, and it would not have been as successful as it was. So just some quick updates for you. My project is wrapping up and I am currently writing my thesis. So the results of our study on Prussian carp in Saskatchewan is that Prussian carp are found throughout the South Saskatchewan River system and its tributaries, and this includes all the way past Saskatoon. They do have the potential to continue spreading in that system into the North Saskatchewan River. We just haven't detected them any further past there yet. The other awesome thing is that the Coppell Dam on Lake Diefenbaker is holding as a barrier to prevent Prussian carp from spreading throughout the Coppell system and further on into Manitoba and the United States. So that's really awesome to know that they aren't in our Coppell system yet, and we're gonna have to fight to make sure that they stay out of there. Also, a really interesting finding is that we actually found male Prussian carp in Stockwell Lake. We expected them to all be females because they are able to reproduce by stealing sperm from other fish. So the fact that there are males indicates that maybe they're actually reproducing normally. So we have now a whole new project on their reproduction that's going to be starting up to continue the research on Prussian carp in Saskatchewan. And finally, I do have my results from the diet analysis of Prussian carp on Stockwell Lake, and we used a technique called stable isotopes to do that. And so the summary of that is that Prussian carp are mostly eating zooplankton and small invertebrates in the water. And actually, juvenile fish are eating mostly zooplankton, and then when they become adults, they're switching to eating mostly invertebrates. So that's just a really quick summary of my study, and I can't wait to share more about it with you in detail. I hope everyone is staying safe and being able to get outside and enjoy the fishing and the hunting in the province, and thank you guys very much.
Hi, my name is Jamie Menard. I'm from Winnipeg, Manitoba, and I'm a first-year graduate student in the biology department at U of R, and I'm currently studying Prussian carp. Current research is going to focus on reproduction of Prussian carp and their spawning habitats, as well as their habits, and potentially if they're spawning by gynogenesis, which would result in triploid females, and that's asexual reproduction, or if they're reproducing sexually using a male and a female to create offspring. Working on the fish at Stockwell Lake, I looked at the sex ratios, and it determined that 83% of the fish we caught that were sexed were female, which indicates potentially a gynogenetic population. But there are males, and so that is quite interesting. And we don't know for sure, but Prussian carp may be reproducing sexually as well as asexually. Prussian carp can take over certain native fish's niches. For example, they overlap with perch habitat and they overlap with white sucker diet. Additionally, we have found them in Cottle Creek and Cottle Bay, and that is an important area in Saskatchewan for walleye spawning, and they can potentially use resources that the walleye need for natural reproduction. I look forward to continuing my research and bringing you more results next year.
Hi everyone, my name is Michelle Chupik. I'm originally from Danbury, Saskatchewan, and I'm part of Saskatchewan Sportfish Research Group. My general interest in science is ecology. I'm especially interested in interactions between fisheries and people. So my master's thesis was on habitat use of fish, fish-eating birds, and people. And my study area is Buffalo Pound, Saskatchewan. From there, I was able to do acoustic telemetry for tracking fish so we can further understand habitat use as well as movement ecology. From here, what my science kind of teased out was fish move around a lot. For the 2019 season, that was especially true for pike. Pike move around throughout the whole lake during the duration of the open water season. And my walleye and burbot really favored the Northwest Basin here for the months of May and June. From anecdotal data, we know that fish do move upstream into the Upper Qu'Appelle River. This is really interesting because sometimes we only think of these fish as lake-dwelling species rather than river too. Especially exciting out here to kind of bring out some more data to see what really drives fish habitat use here, especially for Buffalo Pound, since it's so important for fisheries across Saskatchewan. It is the spot where spawn camp happens, helps stock walleye across Saskatchewan here. It's a really exciting collaboration between Saskatchewan Wildlife Federation, Ministry of Environment, and the Saskatchewan Fish Hatchery. We were able to team up and collaborate as well and collect our fish to track here. It's really great. I'm hoping that in the future we will be able to do more collaborations with Saskatchewan Wildlife Federation. It's been a really great membership here, and really hope to do more stuff. From here, if you like to continue reporting tagged fish, that'll be awesome, as well as keep reporting any suspected invasive carp species. You can email pictures, date, time, location to the email that's on Saskatchewan Wildlife Federations. And we'd like to really thank everyone for helping out, as well as hoping that in future for next year's convention, we're able to meet up in person and show you some more exciting data. We'd really like to thank Saskatchewan Wildlife Federation, and we hope to do some more collaborations in the future. Thank you.